Hi everyone, this is Kevin Wagner, the Keto Advocate. In January of 2016, I had the privilege to sit down and chat with Dr. Brent Reynolds at the first annual conference on nutritional ketosis and metabolic therapeutics held in Tampa, Florida. Let's see what Dr. Reynolds had to say.
Yeah, so I have a fairly diverse background. So my original training is in medical physiology, and when I was a graduate student, I had discovered neural stem cells, and went on formed a biotech company after that called Neurospheres, and we were commercializing some of the things that you could do with the neural stem cells. And then I sort of had an aha moment where I decided that I'd kind of had it with Western science. And so around 1997, I quit doing Western science, went back to school, got my degree in Chinese medicine, and I practiced Chinese medicine on the west coast of Canada for a number of years. And at the same time, my wife and I got really interested in yoga, and we opened a yoga center in Thailand. And so we used to spend half of our year in Thailand at the yoga center, half the year on the west coast of Canada at the Chinese medicine center, of course, the correct times of the year doing that, obviously. And when we're at the yoga center, we primarily did teacher training, so students would come spend about 6 months, live there full time, do yoga, and at the end of it, they get an internationally recognized certificate. And so we hung out with the students a lot, and one of the students was from Australia, and he had convinced my wife and my kids that we should go to Australia because it was like the coolest country in the world. And so my family wanted to go, but I was saying, like, I got a business in Thailand, I got a business in Canada, got nothing to do in Australia. And so at the time, I was also consulting for a stem cell company in Vancouver. And I had invited all my friends from my biotech days to come visit, give talks, license their products to the biotech company. And one guy, a guy named Rod Ritz, who had worked at my biotech company called Neurospheres in Calgary for a number of years, was in Australia. And so I invited Rod. Rod came, had just come back from Thailand. My family wants to go to Australia. And I run into Rod in the hallway, and haven't seen Rod in 5 years. He doesn't even say hi to me. He just says, "Brent, you have to come to Australia. We're starting this new stem cell institute." And it was kind of like, wow, the stars have aligned, right? So I had a reason to go to Australia, went to Australia, and realized I really missed doing academic science. So I got back into academic science in the 2000s and was at the Queensland Brain Institute for about 5 years. And then about 7 years ago, I got recruited to the University of Florida. So my career's kind of spanned going from, sort of hardcore cell biology, going into alternative medicine, Eastern philosophy, and Chinese medicine and yoga, but now coming back around into Western science.
Thanks. Could you tell us about the topic you will be speaking about here at the conference?
So several years ago, we started thinking about, cancer as this multimodal type disease. And we thought that, well, maybe this requires a multimodal type of sort of response. And a lot of this actually came, I think some of these ideas came from my training in Chinese medicine. So I'm also a Chinese herbalist. And in Chinese herbs, you don't take one herb to treat a condition, but rather what you would do is you take a cocktail of herbs. And so each one of those herbs has, literally multiple effective agents in it. But you take like half a dozen or a dozen of them, mix them together, and give it to a person. So we started trying to think along those lines a little bit. And so this got us into thinking about natural products, got us thinking about the metabolic aspects of cancer, in particular brain tumors, and got us thinking about the ketogenic diet. So we've developed recently a polymolecular drug, which is a combination of several natural product extracts. And when we combine that with the ketogenic diet, we get this awesome effect on being able to treat high-grade gliomas or glioblastomas. And the problem was that we had thought at the time, as we started putting people on this polymolecular drug, as we started putting people on the ketogenic diet, we put ourselves on the ketogenic diet and tried it, realized it's really tough to do. And we thought cancer patients will do it because they're desperate. And they'll be willing to go through the pain of the ketogenic diet to get the potential benefit. And that was a mistake. And basically what we found was that cancer patients, because, well, anyone when you're sick, you need comforts in your life, and food is a big comfort. And so cancer patients are sick, having that food comfort is really important to them. So when we realized that sort of error that we had made, we realized we needed to develop a diet that was maybe a little easier but would have the same effects as the ketogenic diet. And so this was when a graduate student, a girl named Regina Marcello, came into my lab, and we were talking about this, and she kind of took this on to develop what she ultimately ended up calling the supplemental high-fat, low-carb diet. And so this is, very similar to the classic ketogenic diet. So, but rather than having sort of a 4-to-1 or 6-to-1 ratio of fats to the other macronutrients, this is pretty close to a 1-to-1 ratio. So it gives a lot of flexibility of what people can eat. But at the same time, what we find, or what we started studying, was to ask the question, does this have the same anti-cancer effects as the ketogenic diet? And so what I'm going to talk about today, so Regina finished her thesis last year. She's got her PhD. She's not now Dr. Marticello, and just kind of go over the data that she basically has. So one of the interesting things, though, that we found was that the ketogenic diet and the supplemental high-fat, low-carb diet, not only does the supplemental high-fat, low-carb diet mimic the ketogenic diet with respect to reducing glucose levels, increasing ketone levels, and reducing tumor cell proliferation. But we also found that the diet is able to target, not selectively, but in addition is able to target the cancer stem cell population. And so Regina did a whole bunch of work along those lines, and I'll present some of that data today. So, what we found is that this, I'll call it the modified ketogenic diet. I'll just get away from the supplemental low-carb, high-fat, which I just tend to mess up. Is that, so this modified ketogenic diet is effective in an animal model. So what we do is we take directly from the surgical suite, we take a patient's tumor. We either put it directly in the animal, or we put it in tissue culture and then put it in the animal. And it basically doubles the lifespan of the animals when we just simply put them on this ketogenic diet. Polymolecular drug that we develop, which we call epidifurfane, we also get a further increase in survival of the animals. So our next step is actually now to move this into clinical trials. In wanting to move this into clinical trials, in the way that we've kind of always viewed this therapy is we never saw this as a standalone therapy. We always saw this as a therapy that would be done together with standard of care. And one of the reasons for that is we think that we can enhance standard of care, reduce the negative side effects of standard of care, and ultimately increase life expectancy in patients that have this disease. The other reason for wanting to do these together is that any clinical trials that get done will probably be done with patients with standard of care. So the odds of being able to go to the oncology community and say, I have this dietary approach, show the data works really well, I would like to take some of your patients, I would like you to stop them or not give them standard of care, and then I would like to put them on the ketogenic diet. The odds of actually being able to do a large phase 3 efficacy trial under those conditions is somewhere between slim and none. So it needs to be done together with standard of care. Surprisingly, you look in the literature, except for Dr. Adrian Schreck, who's just done some awesome work with the ketogenic diet and radiation treatment. There hasn't been any work published yet doing it with the standard of care chemotherapy, which is temozolomide, and there's been no work published demonstrating that it has effects when you use the radiation and chemotherapy together. So really our next step now is to do, in our animal model, is to demonstrate that when you use standard of care together with the ketogenic diet, that you're able to enhance lifespan of the animals that have those tumors, and also importantly that it's not creating any negative unintentional side effects, that things maybe we can't think of that will end up popping up. And third, that it's clearly not going to interfere with standard of care, because that's usually, of obvious concern to the clinicians and of importance concern to the patient. So where we want to, our next step really is to demonstrate that this modified ketogenic diet works together with standard of care, and that'll be the stepping stone to move it into the clinic. So I think one of the things that we've started to conduct, and again we would like to continue to do more of, is combining the ketogenic diet or the modified ketogenic diet together with other low-cost, safe, and effective therapeutics. And so this would have gotten us on along these lines of using natural products because of the low toxicity. And so I'd like to see more studies along those lines. So, one of the, I would call it definitely a discerning issue is the cost of new cancer therapeutics. And so as an example, for women that have breast cancer that overexpress the HER2 receptor, there's been this great antibody called Herceptin on the market. And it's made a real difference in extended lifespan of women. And recently, within the past year or two, they came out with a Herceptin antibody, but attached to a toxin. So the antibody binds just to the tumor cells. When it binds the tumor cells, it takes in the antibody, and then that toxin gets released within that cell. So it's sort of a selective targeting of the cancer cells. And clinical trial data that came out showed it worked great. It gave another boost to enhancing life expectancy. However, the cost of that drug is astronomical. And so I have some personal experience. And my wife passed away a couple of years ago of metastatic breast cancer, was on that particular drug. And so we saw the costs of that. The insurance company was billed $30,000 per infusion of that drug. And you take it every 3 weeks, and you do it for a year. And so this is not an uncommon story. There's a lot of cancer therapeutics that are like that. Insurance companies will not be able to afford it. Countries that have socialized medicine, the systems basically can't afford that. So part of this is also an issue of not developing effective drugs, not developing safe drugs, but also developing cost-effective medications as well. So where we'd like to go is combining the ketogenic diet with things like our polymolecular drug, the epidifurfane, but combining it with other similar types of approaches that are going to increase the effectiveness. And then ultimately what we need to do is we need to move this into the clinic. And so in my biotech days, we had an axiom, which is used a lot in biotech, used a lot in the pharma world, and is that if you're going to fail, fail fast. And so, when you get something, you need to basically go do that definitive experiment. And if it doesn't work, you stop and you go back to the drawing board and you come up with something new. And so the definitive experiment in cancer research is doing a clinical trial and doing a phase 3 trial. So what we're really trying to focus our effort on is taking our modified ketogenic diet together with things like the epidifurfane, that polymolecular drug, or some other sort of approach, and moving that into the clinical trial phase as quickly as possible. So what happened? So when we were going through this process, so what our thought process was on this is most of our models that we use to study cancer, and most diseases actually in the lab, is that they're informative but they're not predictive. So they tell us something about the disease, but rarely do they actually predict what will happen in the clinical situation. So the only way you're going to know what happens in the clinical situation is you need to test it in people in the clinic. So we wanted to get to the clinic as quickly as possible. So that means to get there, rapidly, we need to have something that was going to have low toxicity. And then I think, from my Chinese medicine days and my herbology days, this got me thinking about natural products, about herbs. And so we went in the literature, and there was probably 50, 60, 70 different natural products that have anti-cancer agents, just way too many to study. So what we did is we set an inclusion-exclusion criteria, and we said, number one, there had to be dozens, if not hundreds of research papers, on that natural product demonstrating that it has some efficacy. So we didn't want just a paper that was we didn't want a product that had 1 or 2 papers that were just spectacular, but was that going to be robust? We wanted a robust product. Number 2 is we wanted that compound to already have rodent toxicity data, because we didn't want to take a compound, do the rodent toxicity data, and find there's a problem in it. Number 3, we wanted to have a product that had human safety data already. Again, we wanted to get into that clinical efficacy testing as quickly as possible. We tried to look for products that have what is called generally recognized as safe status at the FDA. Again, just helps us get into the clinic quicker. And then the last sort of inclusion-exclusion criteria, which was actually the toughest to solve, is we had to get a product that was from a reputable supplier. So it had to come with HPLC analysis, certificate of analysis, MDS safety sheets, all that type of stuff, and that we knew batch to batch was going to be consistent. So we did that, sort of did a screen, came up with 6 products. We screened every permutation and combination of those six individual products. And we also screened them, though, at the plasma levels that we know we could get into a human by oral delivery. So we didn't use super high levels of it that we could never get into a person but would show biological effect. We used the levels that we knew could be obtainable in an individual. And so we came up with this unique combination of 3 natural products, which we called epidifurfane, because that's the kind of things my colleagues like. They like names like that. And yeah, so that's basically how we got onto the onto the epidifurfane story.
Okay, and what's included in it?
So there's 3 things. One, there's a catechine that's in green tea. The second thing is curcumin, which comes from the turmeric plant. And you got to be really careful with curcumin because it has really low bioavailability. But there's several entities on the planet that increase bioavailability by modifying the curcumin, and we've tested probably all of those different ways, and we have a couple of products, a couple of companies that we really like. And the third, the bioactive molecule, is a chemical. You find sulforaphane in crucifix vegetables, so broccoli, cabbage, Brussels sprouts. But the highest concentration is found in broccoli sprouts. And so you know the alfalfa sprouts you get at the grocery store? You can find broccoli sprouts. So it's produced the same way. Just take the seed, put it in some water, and you get those nice little sprouts that pop up. That actually has the highest concentration of sulforaphane. So we actually get broccoli sprouts that have been freeze-dried, which keep the active compound in a stable state. And so we combine the freeze-dried broccoli sprouts, the curcumin, the catechine from green tea, and we call that epidipropane. So this was where we wanted to come up with what we may call maybe a more relaxed ketogenic diet. And so the supplemental high-fat, low-carb diet, or call it the modified ketogenic diet, just to make it easier. And what we found is, number one, not only does it mimic the classic ketogenic diet by reducing glucose levels, increasing ketone levels, but number two, it also has the same virtually identical anti-cancer effects. And importantly, what we had found in that study was that it's able to target, not selectively target, but is able to target the cancer stem cell population. And so why this is potentially important is that there's this thing called the cancer stem cell hypothesis that's been around for about 20 years now. There's been a lot of work done over the last decade in it. And what the cancer stem cell hypothesis states is that within cancers, there's a population of cells that has stem cell characteristics. And it's those stem cell characteristics that makes the tumor resistant to standard or to conventional cancer treatments. And so one of those features that a somatic stem cell has, that if the cancer stem cell has, would make it resistant to conventional therapeutics, is this issue of slow cycling. And so, we have stem cells in virtually all organs of our body, and they're ultimately responsible for regenerating that tissue. So as an example, in your blood system or your hematopoietic system, you have a stem cell, which is ultimately responsible for producing 1 billion new cells every single day. That stem cell, even though it is responsible for producing a large number of cells, doesn't divide very often. So it actually divides fairly infrequently. And it may divide, once a month, once a year, something like that. So if your cancer stem cell has the same feature of infrequently dividing, if that cancer stem cell isn't dividing, it's actually going to be resistant to conventional therapeutics, because radiation and most of the chemotherapies that we use selectively target or preferentially target rapidly dividing cells. So the idea here is that what happens when you treat with radiation, you treat with chemotherapy, is you kill the rapidly dividing cells, but that slow cycling cancer stem cell survives. The tumor shrinks because the bulk of the tumor is composed of those rapidly dividing cells. But because the cancer stem cell survives, it then starts dividing again at some point, and the tumor repopulates. So if that cancer stem cell hypothesis is true, targeting the cancer stem cell may lead to better outcomes. And so what we found in the study was that that cancer stem cell population, of which we have a whole bunch of assays that we've developed over the last decade or two to identify that population of cells, we find that the ketogenic diet, or the modified ketogenic diet, does target that population and reduces proliferation. So what we were trying to do when we were looking at the classic ketogenic diet, the thing that makes it very difficult for people to stay on, or at least certainly made it difficult when everyone in the lab went on the diet, was the low carbs. And so what we wanted to do was come up with a diet that would have a carbohydrate the percentage of carbohydrates of your daily caloric intake would be between 10 to 20%. And we found from playing with this in the lab that that is actually a very doable number, and you do have to pay attention to your diet, but it's not like you have to be overly restrictive with it. And so our makeup in the diet with regards to, the ketogenic diet the classic is like a 4-to-1, and so that's, a ratio of fat to versus protein and carbohydrates. And the diet that we used in the study is very close to a 1-to-1 ratio. And so, it's basically 50% fat, and the rest is composed of 10 to 20% carbs and the rest of protein. So, not only is this give a lot more flexibility in dietary choices, may increase compliance in patients, but we've also found that it's also very nutritionally complete. And when we've looked at things like lipid panels things like a complete blood count, things like a complete metabolic profile. Again, doing this in rodents that are on it, is that we find the metabolic profile and the CBC is closer to normal than being on a classic ketogenic diet. So it may be a little bit safer as well, and increased compliance. So not in our hands. And I know there have been a number of papers, there have been very nice papers showing. What we do though is our tumors come directly from the patient, go directly into culture, or they go directly into the mouse. And there's been a fairly large body of evidence indicating that this patient-derived type tumors actually reflect the primary tumor in the human better than the classic cell line. So they reflect the tumor both genotypically but also phenotypically. So when we take our tumors that come directly from the patients and we culture them in a slightly different way than people classically culture tumor cells, we put them into the brain of a mouse, it forms a tumor, you kill the animal, you section up the brain, and you take the patient tumor on a slide here, you take the mouse tumor on a slide here, and you have a pathologist look at those tumors. He can't tell you which one came from the mouse and which one came from a human. Or which came from the primary person. It looks absolutely identical. So whether this is a better model or not, I think still is, is yet to, still needs to be determined. But it is potentially a better model. So I don't know why we don't need to do caloric restriction in our studies, and it may just be that we use a different model so we get a slightly different effect. So we've used a medium chain triglyceride called NeoB895, and it's mostly a C8. So it's a combination of C8 and C10. Can't remember the exact ratio, but it's at least 90% C8, and the rest being C10. So, I guess that's kind of been our experience, and where we were certainly heading with this is, can you get your carbs up to a level that gives you some flexibility and, some satiety, I guess, when it comes to the carbohydrates, but still get the anti-tumor effect. And so from our work, the answer is yes. How high can those carbs go? That I don't know yet, but we know we can be in that 10 to 20% range, supplement with a medium chain triglyceride, and get the glucose reduction and the ketone elevation. If you were using things like ketone esters, you may be able to get your carbohydrate levels even higher. Yeah, so this has basically been, what we've focused on in the last 5 years, where we've developed this polymolecular botanical drug. And, this, really was a focused effort where we looked very carefully at what our options were, came up with the best options, did a fairly broad screen of that, and came up with this unique combination. So the combination that's contained within the epidipherphane, or the EDP, has actually never been used before. It's never been published anywhere in the literature. So it is a unique combination. It's kind of like a new molecular entity, so to speak. And are there other things that may work as well? Absolutely. I think it's just a matter of hunting down those things and testing them in the lab. That's a really good question. I don't think I have a great answer for that. However, I have run across some papers where people have used the ketogenic diet to attenuate cancer cachexia and gotten positive effects. So I guess the little bit I know, I would say no, I don't think those two things oppose one another. So my biggest problem in answering that question is I'm really bad at math. So I think the reason we chose to use the percent of caloric intake was we knew when we think about people, there's obviously, someone's going to consume 1,000 calories a day, and someone's going to consume 2,000 calories a day, and we wanted to keep that percentage, so that was basically standardizing it, as opposed to saying you need to take in 20 grams of carbs as opposed to a percentage of your caloric intake, where then you're going to run into problems. Well, how do you deal with someone who's 100 pounds and someone who's 250 pounds? It just seemed the math would have been easier if we did it on a percent of caloric intake. Well, so what we found is one of the things that Regina had done in this is, at least in tissue culture, she had separated out the effects of the lower glucose and of the ketones. And what she clearly found was that the ketones do target, again, not selectively, but they do target the cancer stem cell population. But they also target the non-cancer stem cell population as well. So it's not like it's a selective targeting. But to kind of answer what I think part of your question is do the ketones themselves actually reduce proliferation of the cancer stem cell? And the answer is yes. Yeah, so we do use an immunocompromised mouse, and that is probably one of the biggest drawbacks of that model, is that those animals have, basically a very bizarre immune system. So you weren't by any chance a reviewer of my NIH grant, were you? Because that was actually one of the aims. So so for high-grade gliomas, when you reoccur, of which virtually everyone reoccurs, is the standard of therapy is Avastin. And so what we're proposing to do is to do Avastin together with the ketogenic diet. Now, not to say that you may get some oncologists and some patients that would be interested in foregoing that standard of care. But again, I think, remember, our goal here is not to do a clinical trial on 10 patients. Is to do a phase 3, a large multi-centered clinical trial. And if the patients are going to be on standard of care, it's going to be easier to recruit and to convince the oncologist that we should give this a try.